So, first of all, I've got research agreements with ASG, Paramed, and Philips, which is relevant later. And yes, I work with Peter Mansfield. This is a picture of him in his office. And people sometimes ask me, what was it like to work with Peter Mansfield? I didn't work with him when he invented MRI. I should make that clear. I came after that point. But this picture shows a bit of that, because he got this building that we work in now built, and then he moved in, and for 25 years, he never unpacked the boxes. You can see the old slides still behind him. And to work with him was to get his full attention, absolute full attention, for a week, or whatever it was it took to finish the piece of work he was interested in, and then he moved on. So your pile of papers would move across the desk as he moved on to someone else. But he got his full attention. That's his ability to concentrate. So I've been very lucky to work at Nottingham, where we've had a lot of different MRI scanners over time. This first scanner I wasn't there for. I didn't do my degree in 1898. But this was Peter Mansfield and actually Peter Morris in the original 0.1T whole body scanner. But by the time I arrived, we had the half Tesla scanner. And the special thing about this scanner was actually it was very good at doing EPI. It didn't do anything else, but it did do EPI. I'll come back to that later. And if you recognise, that's Matthew Clements and Mark Sims. So we went off to work for all the different companies. We got a 3 Tesla in 1992. We got a 7 Tesla in 2005. 2016, we went back down to half Tesla, and I'm going to show you our open scanner, results from our open scanner later. In the meantime, there's been another group within the centre working on development of OPM MEG, and the relevance of these two things here, these two scanners here, is that they're both flexible. So both of these allow people to move around and be imaged flexibly, which is important. And now we're planning to install an 11.7 Tesla scanner in 2026. So it's fair to say I've worked at all field strengths, but that just means I'm old, to be honest. So what are the potentials of these extreme field strengths? So I'm really grateful to people who've given me slides here, and the first slides have come from Nicolas Boulant in Paris, who's got these amazing results recently showing the power of 11.7T, the very first 11.7T images from the scanner. And you can see here the sensitivity and increase in contrast in the vessels, the veins, which we're familiar with at 7T, and more so at 11.7. And really interestingly, the increased contrast, directional dependent R2 star relaxation in the myelinated tracts. And I noticed there was a poster here looking at lower fields at the variation in this contrast with field strength. So the scanner gives good susceptibility contrast. Obviously, its capabilities have yet to be explored. It's very good imaging of the cerebellum. The problem with this scanner, the problem with all these high-field scanners, and the problem with most conventional MRI scanners is it's really big. This one's really, really big. There's the bore in there. And so this means it's not flexible. You know, you can't take it to your nearest car park, obviously. In the meantime, Andrew Webb provided me with these incredible images from 0.05 Tesla, 50 millitesla. So you can see here really exciting images at different contrasts. You know, it's capable of doing all sorts of contrasts. No denoising or filtering applied here. He modestly called them the successful cases, but I'm sure he's not the only person to give out successful case slides. This is 234 times lower field strength than the ones that we just saw. I would note, I was just given these slides by Andrew and Nicholas, and I calculated the voxel size difference. And the difference in voxel size is 500-fold in terms of volume. So I think we can say, to the first approximation, given that there was no competition going on here, it's approximately linear scaling with voxel size. But do we always need these tiny voxels we get at 7T or 11.7T? Will some bigger voxels do? You can get different contrasts at low field as well. And the thing about the low field scanner, as Andrew's very well demonstrated, is it's extremely flexible. It can be moved around anywhere with all these low field scanners. So in the meantime, we've got this open MRI scanner. It's not mobile, far from it. It's very heavy. But it is allowing flexible imaging, and we got it for several reasons. So first of all, obviously for patients, they can sit up in it. It's kind of less claustrophobic, allows rapid positioning. this is a study by Rob Deneen and Caroline Costigan, who were aiming to look at whether children were more easily scanned in the upright scanner. And they certainly liked the scanner. They preferred the scanner, but it turned out that they really, really liked as well looking at videos and the new Philips scanner, which was designed to be very good for children, that we have as well. So it turned out that there wasn't much benefit clinically. In terms of weight bearing, I'm not showing weight bearing joints particularly here, but the spine. I mean, anybody who's done MRI is not used to seeing a spine looking in that shape. You can flex the spine in this scanner. This is a scoliosis spine. Anybody who's got an interest in scoliosis and wants to look at it in MRI, I feel this is a missed opportunity. We're very keen to look at that. And of course, you can use it for looking at human dynamics in an upright position. I'm going to come back to this a lot later in this talk. But these scanners, the flexible scanners, particularly the scanners that have the open bores, can have worse performance. So it's a C-type magnet with a 56 centimetre gap, which we're up against sometimes. It's got a 30 centimetre sweet spot, so to speak, and that is limiting for people with big lungs. But the key thing is the gradient performance, because it's got these flat gradients as opposed to the cylindrical gradients we're used to. The gradient performance is worse. And the question is, you know, how does that limit us? And we've been looking recently at various ways of compressed sensing and field correction to try and overcome some of these problems. But somebody has been looking at imaging speech in the 3 Tesla scanner. And I should say, there's been similar work doing this at Half T from Christian Nyack. But this work was done on our 3T, and somebody said, I want to do it on the upright. And I said, it'll never work. It'll never work. The gradients aren't good enough. We're going to need to get a physicist in and spend time developing this properly. So last week, they tried it and proved me wrong. This is at 10 hertz and 1.5 by 1.5 by 10. This is at 4 hertz and 3 by 3 by 10 millimeters, sorry, I should say. So clearly there's trade-off here. It may be this isn't fast enough, but you know the scanner has pretty good performance. So the question is, which scanner for what? So I'm an experimental physicist and I teach undergraduates, and this is a phrase I use a lot. Your data needs to be good enough, but not better. As you go better than good enough, you're just wasting effort, or in the case of this talk, wasting electricity possibly, wasting CO2 emissions. And so you need good enough, but not better. So the data needs to be acquired for long enough, but not longer. Now, I'm not a clinician, I'm not a radiologist, and I'm in no way qualified to talk about what's good enough in radiology, and that is a big question. I know radiologists, you know, don't like physicists making things too good. But these scans are, this is a news item from Science showing the installation of a low-field scanner in Uganda. And these are images from the same scanner, hyperfine scanner, I believe, at Yale, comparing a conventional scan and the low-field scan. And you can quite clearly see the hemorrhagic lesion. And in fact, you can see detail within it. That scan's good enough to tell you it's there. If you want to look at whether your tumour, a tumour has been resected properly, whether it's recurring, you may want conventional scanning with high resolution and contrast agents and things like that. So it's possible, well, I'm sure it's the case that for some circumstances you need high quality scanning. Maybe for screening, low quality scanning to work out why someone's got a headache will be enough. I'm not going to show any 7T data today because I'm sticking to sort of dynamic things for the end of the conference, but I just thought I'd better show some 7T data. So this is comparing magnetization transfer measured at 7T, which is very sensitive to MT if you can overcome the SAL problems, and functional connectivity from MEG with structural connectivity based on the MT scans from 7T MRI. So this is, you know, again, if you want to do really detailed neuroscience, you might need higher fields. Now, actually, Jose talked about this quote on Monday, at the beginning of this meeting, and I'm not sure I can use the mouse. I don't know if the projectionist can play this sound here. When Peter Mansfield died, his granddaughter was talking about him to the media, and she said one of the things that he'd said was that he didn't really feel that, when asked about how many people's lives he saved with MRI, he said, well, I'm not sure that's how I see it. What I remember is that when people got that MRI diagnosis, it changed their lives, and often for the worse, because maybe there was no treatment. But I think he actually knew, and he certainly inspired me to see MRI in a different way, because the way he got me to work with MRI originally, when I first arrived to an EPI-only scanner, was to use it as a dynamic functional imaging technique. Right from the beginning, right from 1990, we were using MRI for experimental medicine studies. So MRI is a window into physiology and biochemistry. It's safe, non-invasive, repeatable measurements for experimental medicine and experimental bioscience. So there's two ways, or many ways, you can use MRI in medicine. One's for diagnosis, the other's for guiding treatment in an individual, maybe for interventional studies looking at delivery of a drug in a tumour. But you can also use it in experimental medicine, experimental biology studies where you actually investigate how a drug works or investigate how a disease process works to learn about medicine. So this is another way of using MRI. And that's kind of where I'm going to focus quite a lot today. So dynamic responses to physiology and physiological challenges across organs can be studied with MRI. You couldn't do that with X-rays or CT. And even if you could, you wouldn't get the wealth of contrast mechanisms you get with MRI. So we look at the brain, we look at the body, we look at the interactions between them. I saw a great talk yesterday afternoon on the brain-body aging, using the biobank data. This is, you can see here the heart, the stomach, the blood vessels in the liver, all the wriggling around in the small intestine. I'm not going to talk about spectroscopy today, but of course you can do spectroscopy. You can do studies, for instance, looking at liver response to a fructose challenge. It depletes the ATP, and the rate of depletion depends on your BMI, which tells you about the energetics and the mitochondrial function of the liver. This is the sort of thing you can do with MR at high field. I'm going to now focus on three areas. I'm going to focus on the gut, the lung, and the placenta. And I have to say, the gut and the placenta, Peter Mansfield, well, certainly the fetus, Peter Mansfield started me looking at those when I first arrived at Nottingham. So it's been a career based on these, really. And we use MRI to look at the gut. And this is a good example of how MRI can, it obviously can't do everything, but it feels like it sometimes. When people have functional GI disorders, which are frankly very underserved in clinical practice, at least in some areas of the world, they may come and have manometry. They put a tube into their gut and measure pressure changes. They may have pH measurements, or they swallow tablets that have radio transmitters on them. They may have barium enemas or barium meals. They may have gamma scintigraphy studies to look at food transit through their gut. Multiple studies in multiple different mechanisms, and quite a few of them using X-rays. So MRI can study all of those things and more. So up here again is the image of the stomach. And if you look at this stomach, it looks like it's pumping contents out. This is a coronal image, and it looks sort of J-shaped. Actually, if you look down here, we've got the motility of the stomach marked with tagging. You can also use PCA. And you can see here that these tags are moving both ways. And this tells you that down in this part of the stomach, in the antrum, there's retrograde flow, which is causing mixing, breaking up of food products, and indeed emulsification. Back on the old half-T scanner, this is at half-T on this EPI scanner, we could look at the the dilution of a model's meal in the stomach as it became more diluted over time. And this wasn't known. It would take, over an hour for this meal to begin to dilute uniformly. It dilutes from the outside. That wasn't understood, and this is important for drugs. On the old half-T, we gave beads that we looked at them breaking down in the stomach to measure forces in the antrum, and now we're doing this on the 3T. If you swallow a meal, a meal that contains fat and water, the fat will will float on the water contents. And in fact, one of the first scans I ever did at the Marsden, I remember seeing my mother, I was scanning my mother, she just had fish and chips. I remember seeing the level of fat from the fish and chips to the Royal Marsden Hospital. Anyhow, you can see, you can't quite see on the screen, I think, but there's water there and there's fat layering there. This matters because if you're sitting up, it means the fat layers at the top of your stomach and the water is delivered, or the watery content is delivered to your duodenum first. When the fat hits your duodenum, the stomach stops emptying, and you will feel full if that fat stays. So if you eat an emulsified meal, you will feel full. And in fact, we have done a study that shows you should eat soup if you want to feel full, because the soup emulsifies the fat. We've looked at foams to increase satiety, a lot of work on looking at effects of satiety, rafts to reduce reflux. But you can go on and look at the bowel, go on down. So over here, we're looking at the small bowel movement in healthy subjects and in cystic fibrosis. And actually, I should mention, this is Motorland, a company in London, who've actually done the analysis, the registration of this data. And here's the bowel again. I don't know if you can see the mouse. Here's the bowel again. And this is the healthies, and these are the CF, and there's very distinct differences in the CF. And this is because they've got dysbiosis, which is a change in the microbiota in the bowel. And over here, we looked at whole gut transit. Luca Marciani's got a product that you can now swallow to provide gut transit markers. There's a similar thing for X-ray, but obviously this is commonly used in children. We don't really want to be using X-rays in children, dynamic X-rays in children. However, that's all about the lumen. What about the bowel wall? So the lumen, we do it at 3T because we've got a 3T scanner, honestly. We have 1.5T, it's quite old. We want to move some of this to 1.5T because we think it's good enough, 1.5T. We don't actually have any problems with 3T, I should say, with RF and SAR, but we're not ones that we don't work around. But we want to look also at the bowel wall. So perianal fistulas are sort of self-explanatory, and they're not uncommon in patients who've got Crohn's disease. These are young people, this is sort of devastating for them. And again, it's an underserved area of medicine. They're currently monitored at 1.5T with contrast-enhanced MR. So the study here was looking to see whether you could replace the contrast with quantitative MRI, in particular MT at 3T, and indeed it appears you can. We've also been looking at other signs of gut wall inflammation in Crohn's disease. So one of the things that happens in Crohn's disease is something called the comb sign, which is where you get hypervascularization. And so we've looked at using basically a Dixon scan to pick out the vessels and look at the vascularization against, in this case, BMI, and its changes in Crohn's disease. And this is 3T data, and I think you'd need 3T for this. So this work has been extended to look at this bowel wall. So this is looking at the bowel wall in patients, not patients with Crohn's, this is healthies, being given a lactulose mannitol test, which changes bowel permeability, and seeing a change in T2 in the small bowel wall at 3T. And then in patients with Crohn's disease, looking at the MTR of the fibrotic lesions in Crohn's disease. The lesions are thicker, so it's easier to measure. But I think you probably need 3T for this sort of quantification. And we've also done it ex vivo. At 7T, this is collagen at 7T ex vivo, just in a test tube. And you can see very clear CEST effects. This is a CEST spectrum. So this is looking at water exchange between molecules in different components. And you can see here that the water exchange shows very prominent effects of this collagen. This is really quite prominent effects of the collagen. And it's actually affected by cross-linking. So we haven't done this at 7T in vivo, but it would be a good place to go to look further at these fibrotic areas. But who eats lying down? Well, the Romans did apparently, but we are of course interested in looking at gastric emptying in the upright scanner. These are just a couple of quick scans showing, in this case, porridge. You probably can't see it actually, porridge in there being digested. And this is one of the first images we got in this scanner, and it made us realize something. So this is somebody swallowing water, and you can see their stomach getting fuller as they swallow water, well, actually orange juice, as the stomach gets bigger. And this is somebody who's swallowed orange juice. And what we found in a study of about 10 subjects is in some people, their stomach goes from here to here when it's full. Some people it doesn't. And we had no idea from that, from supine MRI. You know, you just don't know. And in fact, from any other technique, I think quite a few of the others are done upright, lying down. So once we did supine MRI, we realized there's a sort of extra dimension of gastric function we were unaware of. There are other areas of the guts we're interested in. So this is a coil Paul Glover built because the RF coils need different designs on the the transverse field of the upright scanner. So on the upright scanner, the field's going this way across the body if you're sitting facing the front, whereas on the conventional bowls, of course, it's going through from head to foot. So this changes the RF coil design. So we built a coil that was basically, it's called the potty coil, but basically there's a commode in the scanner here, and this coil sits around the commode. So we're able to look at proctography, which is something that's otherwise done with X-rays. And this is in a healthy subject. We've done it in a series of healthy subjects at the moment, and you can see clearly they're able to image evacuation of the bowel. Now, this isn't a funny subject, but I had to put up this image here because I saw it. Apparently, this is a local character in Barcelona, but the point actually is not so funny because this is related to pelvic floor issues, which are quite, again, an underserved area of medicine. So now I'm going to move on to talk about lung function. So, yes, I don't know what the time is now, sorry. So I'm going to talk about lung function, which is an area when I started, I would have thought was impossible to study with MRI. And I must admit, you know, I was skeptical again when we started on this journey. So there's been a lot of work recently looking at lung function with a technique called pre-full, which is basically the simplest MRI scan in the world. It's simply an FFE scan run quickly as a dynamic series so that you can then monitor the signal in the lung during breathing. So you monitor respiration, where have I gone? Respiration from the navigator ROI, trivial. So you can get a measure of the lung volume effectively, and you measure the parenchyma signal in the voxel. And what Zach has been doing in our lab is a slightly variation on pre-full, so correlating the signal from the parenchyma with the signal from the lung. And in the simplest case, the curve will look like that. There's a linear relationship and a voxel, but actually it can be much more complicated and more interesting in what's going on in these curves. So you can get the correlation coefficient. This is in a healthy, this is a patient with COPD. You can get the gradient. You have to be careful how you calculate the gradient, but basically you find some negative areas where the gradient's going the wrong way. So when you breathe in, you expect the signal to get lower. In some cases, it goes the other way. And this is more exacerbated in patients. But basically, you can see differences in this correlation coefficient between healthy smokers and then patients with COPD and other conditions, and similarly with the gradient. So, of course, you don't tend to breathe lying down, so we went to doing upright MRI for this. First of all, there was an interest in looking at the diaphragm and looking at the way your diaphragm varies when you're lying down and sitting and lying down, and when you're breathing in and breathing out, measuring the volume under the diaphragm effectively, and the excursion of the diaphragm. And the aim here was to look in patients at changes. I would say we haven't proved it different in patients, but the point is this study looking at diaphragm function is important. And it's one of the treatments to change diaphragm function in COPD, but they can't lie down to have a conventional MRI scan. So I have to say, this is probably another case of me saying this won't work, and it did. So Arthur Harrison then said, well, we were going on to try and do lung imaging at half T because, of course, the T2 star effects are less at half T, so we did expect it to be better. So in this case, we had somebody lying down and repeated the same study. This is conventional pre-full analysis. So you can measure the ventilation cycle, the fractional ventilation, and the time to peak of the ventilation. And in fact, you can do the same for perfusion. I didn't show that in the previous slides, but by correlating with the cardiac signal in the aorta, you can look at perfusion signals. So you can do that lying down. Of course, you can do it sitting up. And Arthur was very keen to do it upside down. And you can do that, of course, in the scanner quite easily, provided you've got volunteers prepared to do it. And it's an interesting result, really, because if you're sitting up, if you look at the difference between the apex, the middle, and the base of your lungs, the top, the middle, and the bottom, there's a difference in the fractional ventilation. So the ventilation is most at the base, which I guess sort of feels reasonable, really. And that's largely the same if you're sitting or if you're supine. But once you're head down, upside down, it falls, it evens out. That change doesn't occur. And actually, it's similar if you do deep breathing. If you look, this is normalized, so it's now normalized, the average ventilation across the lung. If you add in the flow, the Q part, that's the blood flow, you sort of see something similar in the sitting and supine conditions, so that it looks like the flow and the ventilation are matched. But once you turn upside down, there's a bit of a mismatch maybe beginning to appear here. So it looks like gravity influences lung function. We're used to breathing in an upright position or lying down position, but there seems to be a bit more going on. So when you turn upside down, it doesn't reverse. It looks like your lungs have grown that way. And there seems to be a flow ventilation match that's kind of okay sitting up and lying down, but changes when you're upside down. Right, and finally on the MRI, I'm going to talk about placental function, which is one of my favourite areas. So this really old data set is a really important data set, which was collected in the 1930s, I think, in England. And it was a midwife who collected the weights of the baby and the placenta, and she kept extremely good records. And this data was rediscovered in the 90s by somebody called Barker at Southampton, and he realised there was a relationship between placental weight and birth weight and actually fetal outcome in midlife. So people who are more likely to have metabolic disorders in later life if they'd had a low birth weight. So it's really important to study placental function, because the placenta is what causes the baby to grow ultimately. So this is a placenta if you don't know about it. I tend to call it inside-out lung. There's blood coming from the mothers through spiral arteries like here. And there's fetal vessels in sort of trees here. So the fetal vessels hang down in a bath of maternal blood. And the maternal blood squirts in and then finds its way out. And there's a whole story about how it finds its way out. We won't go into that today. So this is half-T data from a long while ago. You can see this image and this image. This is the fetus, the placenta, and this is the fetus. And the difference between these two images is it was ASL. So this was uninverted. It was non-selective and selective inversion. It's fair. Here's a half-T scan, perfectly adequate. And here's a 3T scan. And we use 3T partly for reasons I'm going to show. We're interested in looking at oxygenation. But for looking at fetal brains and clinical scans like that, I would defer to others, but I imagine 3T is better if you want to look at the brain. But you can see this big black blob here. That's due to RF interference. The fetus and the amniotic fluid are highly conducting, so you get a very big RF interference at 3T. You can steer it around by adjusting the RF. So, we're interested in the placenta. We moved away from the placenta, but it is a problem. So this is data from Jana Hutter also at half-T. Let's just compare this one with the last one. So that was half-T data from 30 years ago, to be honest, probably. And this is half-T data from today. It's from the 0.55 Siemens scanner, showing that really, honestly, they're amazing images. And I think she might disagree with me when I say maybe fetal brains are better scanned at 3T. So you can get really fantastic data at half-T. There's lots of quantitation you can do on the placenta, because it's actually the fetus is a very well-matched organ to scan on MRI. It's very well susceptibility matched. So we use IVAM to study, so diffusion-weighted scanning effectively, to study flow. And you can measure the apparent diffusion coefficient of the fast diffusing component. and use that as a measure of this fast flow from the placenta into the placenta. This is controls, this is preeclampsia. What we found was that the data isn't actually very bi-exponential. I'm going to come back to this sort of bounce in a minute. We fitted it to a ketosis model, and we found differences in IVAM in compromise. These are patients with preeclampsia or fetal growth restriction. That's when the baby's growing slower. And also in ketosis. But more recently, Jana has also shown that you can do ADC on the 0.5T Siemens scanner very well and get similar results. We've been looking in more detail at this data. So IVAM uses a model which assumes that the vasculature of the capillaries is random, so that you can model random movement through the capillary network as a random walk. Now, actually, the original paper by Le Bihan from 1987 actually mentioned that, of course, flow like that isn't like that in the body. And if you had something like this, which looks not unlike the spiral artery squirting into the placenta, so a curve like this, a whirlpool, you're going to get variations in velocity across this voxel, which will give you dephasing. But you're not going to get dephasing in the way we expect for diffusion, because at some point later, it might bounce back. You're going to get flow-induced echoes. And that's what we see. So I did sort of point out on the previous slide, this is data, real data. We can see echoes, which are way above the noise. Here's more real data showing a bounce. Here's data showing bounces. We've used a mathematical model to model the flow in the voxels, and also shown that you will get these sorts of effects, and you can kind of predict when you're going to get them, and indeed they are adjacent to the spiral arteries. So this gives us more insights into the placenta. Finally, you can measure flow. I'm going to speed up a little bit, but you can measure flow. The flow is very slow in the placenta, which makes sense because if you measure the oxygenation is slightly deoxygenated compared to oxygenated blood. This is measured with susceptibility, but it's not very deoxygenated. And the difference between healthy controls and patients is that in healthy controls, the oxygenation is actually really well behaved, and it's got neat patterns of high oxygenated and deoxygenated areas, whereas in the patients, it's much more scattered. And I think this suggests there's poor perfusion through the placenta. You're getting deoxygenated and oxygenated areas, highly oxygenated areas, suggesting a bad mismatch between delivery and use of oxygen. And again, we can measure oxygenation with susceptibility. We can measure things related to oxygenation with T2 star. And again, Jan has shown you can measure T2 star really well at half T. I think the low flow makes sense because the oxygenation difference between the mother and the fetal blood supplies is quite small, necessarily, if you think about it. Otherwise, you're going to end up the oxygen maybe going the wrong way. So the oxygen delivery gradient is small. So in that case, it's not like in the brain. You only need slow flow to actually get an efficient transfer. So finally, MRI can find new things. So we've been looking at basically dynamic data series of placentas. And this, this is, this is a placenta here. And this is what is normally called a Braxton's Hicks contraction, I think. So the placenta and the whole uterus sort of contracts, and there's a signal change in the placenta. But what we see a lot of is this, where the placental bed contracts, and the rest of the uterus actually squeezes outwards, gets bigger. And we've called this a placental contraction. When we first saw it, I was rather surprised, but actually it turns out that biology predicted it. So there's quite a lot of biological papers showing there are contractile elements in the placenta, that these villus trees have contractile elements, and that they would pull the placenta together. So it's not entirely surprising we're finding contractions. And at the moment, we're looking in more detail at how the contractions work, and are there differences? There do seem to be small differences between high-risk pregnancies and healthy pregnancies, probably related to the, I think it's probably related to the relaxation of the placenta after the contraction. So, just really briefly, I was also asked to talk about sustainability. So I've been thinking about this. Now, you might have noted at the beginning on the slide Josie talked about, I'm actually a local councillor, so I spend a lot of time thinking about this. And I'm very fed up of pictures like this because, you know, every council in the country, in the UK, has declared a climate emergency but done nothing about it. So I'd rather see pictures like this if we're going to talk about sustainability. So there's a number of ways we can talk about this in terms of MRI. In terms of the scanners, I'm going to bandy around some numbers which I didn't write on the slides, and they're all real approximations. But the sort of quote is 20 kilowatt hours for an MRI scan. So the hyperfine scanner says 0.7 kilowatt hours for one of their scans. There are ways the manufacturers can improve this, the ways they can turn off the scanners at night, and of course they're all doing this anyhow, partly because otherwise you need a bigger power supply to actually put the scanner in. This is from NearScan showing the high temperature superconducting materials, which makes the scanner more efficient. I won't go into details, but it's more efficient in terms of the energy required to cool it, and also in terms of the fact it can be reconfigured and the materials can be reused, which I've seen magnets taken away to be put into landfill, which is really sad. So our scanner needs, I hope I've shown, to be good enough, and sometimes 1.5 T is good enough, sometimes half a T is good enough, sometimes 0.05 T is good enough, and sometimes we need 11.7. I saw yesterday somebody talking about, well, lots of people talking about, about MR0 code, and somebody talking about correcting their sequence on the code on the simulator before they went to the scanner. And I think we can learn a bit from radiation where it's very important to get the scans right first time. Data processing, there's a lot of work talking about going onto the cloud. This is small compared to the scans. We're talking about sort of 0.03 kilowatts for storing MRI data, I think, but the kilowatt hours for storing MRI data. But it's this amount of kilowatt hours to put a gigabyte of data on your hard disk. It's this amount to put it on the cloud, apparently. These numbers are all very approximate. So basically, saving 100 hours of video on the cloud is equivalent to a flight from London to Barcelona. However, the biggest shock, which I've seen recently from a paper from Manchester, is that the biggest impact is actually travel to the centre, which obviously is a reason to have a low-field scanner and take the scanner to the patients. This is a scanner car park. This is the hospital car park at Nottingham, or one of the many hospital car parks in Nottingham. We have, and I'm sure many of you have developed methods of running your scanner remotely during lockdown, and we now run some of the scanners frequently remotely. And we certainly plan to run the 11.7 that way so people in the rest of the country can use it without necessarily visiting the centre. It turns out, apparently, I'm told, 6 kilowatt hours for 20 miles in an electric car, which is a big fraction of the cost of the MRI scan. So, in conclusion, low field is cheap and sustainable in terms of kilowatt hours or CO2 emissions. It's potentially very accessible, much easier to reach the patient. It's portable and flexible, low SAR, low acoustic noise, which we haven't mentioned, and lower power. High field is useful for things that demand high resolution or high sensitivity. So at the moment I'm doing a study looking at the TRN, a needle in the haystack of an area of the brain, which you can only do at 70 really. Spectroscopy, multinuclear spectroscopy, you need really high field. ASL benefits from high field. CEST, the chemical exchange, benefits from high field. Flexible scanning usually has lower spec, but I haven't mentioned today that it's very sensitive to motion. So here's some scans, and this is Peter Mansfield with the ex Prime Minister of the UK. The reason I bring it up here is he did have a really good insight into the power of MRI. So one of the very first talks I gave was a very embarrassing talk, talking about injecting gadolinium into a melon because he had a vision of using MRI for interventional radiology. So in conclusion, the scanner needs to be good enough and no better. And MRI can provide a non-invasive way of monitoring physiology and biochemistry, and the art's in designing the quantitative experiments to probe the accessible biology. And I want to thank lots of people, but I've got to mention Olivier Mugan, who's really key to the half Tesla and the 7 Tesla project in Nottingham, Luca and Caroline for the gut work, George and Amy for the fecal work, and Arthur and Chris for the speech and the lung work on the upright scanner, and Zach and Sue for the lung imaging. Thank you very much.
Thank you so much, Penny, for this absolutely fascinating and inspiring lecture. It is now time to move to the next part of our session today, which is the